Hi, everyone. Thanks for joining. My name is Drew Heinmiller. I'm the product manager for our photoacoustic line of products here at Fujifilm VisualSonics. And I'm happy to present today Dr. Martina Capozza from the University of Torino. She's going to be talking about novel contrast agents for photoacoustic imaging of cancer. A few things before we get started. We are recording this webinar, and a recording will be made available afterwards. So if you're not able to stay on for the whole thing, you can catch it later. All of the lines are muted for the duration of the webinar. If you have any questions that come up during the webinar, we will be having a question session at the end. So please type your questions into the questions box on the side panel there, and we'll try to go through as many as we can at the end. This presentation will be anywhere from 30 to 45 minutes, and then 10 to 15 minutes of questions at the end. I'd like to introduce Dr. Martina Capozza. She received her MS in molecular biotechnology in 2013 and her PhD in pharmaceutical and biomolecular science in a collaboration between Bracco Imaging Research Center and the Center for Molecular Imaging at the University of Torino. She defended, I should say, in March of 2017. Her thesis was titled Nano-Sized Systems for Imaging of Focus on Photoacoustic Imaging. And from May, she's a postdoctoral fellow in the Laboratory for Preclinical Imaging at the University of Torino. And she's pursuing her interest in photoacoustic imaging and the development of novel contrast agents. And that's what she's going to be talking about today. And she's talking about these some ICG-based contrast agents, nanoparticles and dyes for targeted molecular imaging today. And I'm going to hand it over to Dr. Capozza now. Okay, take it away.
Okay, thanks Drew for the kind introduction and for the opportunity to share my results about novel contrast agent for photoacoustic imaging of cancer. So, human cancer frequently displays substantial intratumor and intertumor heterogeneity that can affect therapy response. The aim of this research project that I present today is to develop a new methodology to evaluate molecular and physiological features of tumor to characterize tumor heterogeneity. The ideal technique to reach this goal should be characterized by a good spatial resolution, good penetration depth, good sensitivity, and but also should be cheap and involve the use of non-ionizing radiation to allow repeated imaging acquisition during time. The technique that we select to perform this study is photoacoustic imaging. That is a new imaging technique that combines the high contrast and spectroscopic-based specificity of fluorescence imaging with the high spatial resolution and penetration depth of the ultrasound. Compared to the other imaging modality, photoacoustic imaging is characterized by a good spatial resolution, good penetration depth, and good sensitivity. Another advantage is the use of non-ionizing radiation. Okay. Photoacoustic imaging is based on photoacoustic effects. So basically, we have a pulsed laser beam that irradiates tissues of interest. Then we have absorption of photon that cause a thermoelastic expansion that generates pressure waves that are detected by an ultrasonic transducer. Thanks to the presence of a tunable pulsed laser, we can perform multispectral imaging. We can perform multispectral imaging of endogenous chromophore like oxy and deoxyhemoglobin or melanin, and we can have information about tissue epoxia or vascular volume, as shown in this preclinical application or in this clinical application. In this clinical application, the signal of hemoglobin was used for diagnosis of active Crohn's disease. The introduction of exogenous contrast agent can improve specificity and sensitivity, and we provide new tools for the evaluation of tumor heterogeneity. But an efficient photoacoustic contrast agent should have a high molar excision coefficient in order to maximize the amount of light that is absorbed and low fluorescence quantum yield to maximize the amount of energy that is converted to heat, and for sure high photostability. For example, in this clinical application, ICG has been injected to detect malignant sentinel lymph node in melanoma patients. All these applications demonstrate that photoacoustic imaging has enough penetration depth and spatial resolution for clinical translation. So, in this research project, in order to improve biodistribution and tumor uptake properties of ICG, we proposed two different ICG-based contrast agents, one for passive targeting and one for active targeting. But we've also investigated different, so the optical and photoacoustic properties of different small molecules near dyes and also gold nanorods. Of all these contrast agents, we evaluate in vitro the main properties that influence the generation of photoacoustic signal, such as molar excision coefficient and fluorescence quantum yield. The photoacoustic signal is also investigated in vitro in different media by means of an agar quantum. Based on the above parameters, a subset of contrast agent is identified and tested in vivo in healthy mice and in tumor-bearing mice. We also perform optical imaging to validate the results obtained with photoacoustic imaging. So these are the ICG-based photoacoustic contrast agent. So ICG is an efficient optical and photoacoustic contrast agent, but it suffers of low photostability and fast blood clearance. So in order to improve the biodistribution and tumor uptake properties of andocyanin green, we developed these two novel probes based on ICG for tumor photoacoustic imaging, exploiting on EPR effect and molecular targeting mechanism. So ICG was coupled to an integrin binding vector, in this case the cyclic RGD, to try a molecular imaging approach, and ICG was loaded on these solid lipid nanoparticles for passive targeting approach. So in the cyanin green, this is the synthesis and characterization of ICG RGD. So ICG was conjugate to the alpha V beta 3 targeting molecule, and the reaction product was purified and characterized. We also observed that the optical properties of ICG are not affected by the RGD binding. Indeed, the absorption and photoacoustic spectra are comparable between the ICG and the ICG RGD in both in buffer and also in seronor. But also the fluorescence quantum yield was not affected by the presence of RGD both in buffer and in seronor. The solid lipid nanoparticles, so we load ICG on solid lipid nanoparticles. This nanosystem are characterized by a solid lipid core. We have phospholipid around, and they are pegylated, so in order to avoid macrophagic uptake. We load these nanoparticles with different imaging probes, like fluorescent molecule or gadolinium complex for MRI experiment. The fluorescent molecule in this case is ICG, and these nanoparticles have a diameter of 15 nanometers. They have a negative zeta potential, and we observe that after the loading of ICG, the incorporation of ICG in the solid lipid nanoparticles enhance the optical properties of ICG. And we can confirm these results via absorption measurement, but also via this photostability of measurement. We also observed that the loading of ICG in the nanoparticles increased the fluorescence quantum yield of ICG. This is not optimal for photoacoustic application, so in order to decrease the fluorescence quantum yield of ICG, we increase the amount of ICG loaded in the nanoparticles in order to decrease the fluorescence quantum yield of ICG. So we optimize the amount of ICG that should be loaded in our nanoparticles for photoacoustic application. Beyond ICG, we also explored the optical and photoacoustic properties of other photoacoustic contrast agents like cyanine 5.5 and others. Among all the tested dyes, there is also this asymmetric cyanine that is an IR dye quencher purchased by Lycor for FRET assay. Since this molecule don't show any fluorescence, we decide to test this molecule for photoacoustic application. So the first step in the evaluation process of novel contrast agent for photoacoustic imaging is the characterization of their optical properties. So the measure of molar excision coefficient and fluorescence quantum yield. Of some dyes, we also evaluate the fluorescence quantum yield in serum and in buffer. From this data, we select those dyes that show the higher molar excision coefficient and the lower fluorescence quantum yield. Another contrast agent that we investigate are non-spherical gold nanorods purchased by nanohybrids. They have an excision coefficient several orders of magnitude greater than that of small molecule dyes due to a localized surface plasma resonance phenomenon. They present a maximum, or they also present a maximum absorption at 800 nanometers. In order to measure the photoacoustic signal in vitro, of this contrast agent, we optimize an agar phantom. This is 2% agar phantom with polyethylene tube inside, so we can load our contrast agent in this tube, and we set India ink as a reference. We select agar phantom in order to have a refractive media for our phantom. So these are the in vitro photoacoustic signals. We can observe that the most we measure the photoacoustic signal in buffer and serum in order to evaluate the photoacoustic signal after albumin binding of our contrast agent. And we can observe that in this case, the photoacoustic signal at the maximum absorption wavelength of each dye was divided by the reference India ink solution. Then normalized data were plotted versus the dye concentration. ICG, RGD, and ICG showed a high photoacoustic signal in buffer, but in serum the signal decreased substantially, and this is probably due to their strong interaction with plasma protein and consequent increase of fluorescence quantum yield. But when ICG is loaded in as a solid lipid nanoparticle, the photoacoustic signal was only slightly affected by the albumin binding. This can be due to the fact that after the loading of ICG on the solid lipid nanoparticle, the albumin binding can be relatively prevented. For the quencher and for the gold nanorods, the signal was high in buffer and not drastically affected by the presence of serum. This can be explained considering that their fluorescence was very low in hot media. So in buffer and also in serum. So in buffer, we can observe that the most promising contrast agents were ICG and ICG-RGD, but ICG solid lipid nanoparticles and the quencher were the most promising photoacoustic contrast agent in serum. In the end, we can observe that the signal of ICG solid lipid nanoparticle was double compared to the signal of ICG in serum. After this in vitro evaluation, we perform in vivo first preliminary experiment in healthy mice. We intravenously injected ICG, IR783, ICG solid lipid nanoparticles and gold nanorods in healthy mouse. And we observe the blood clearance and the biodistribution. In order to observe the blood clearance, we perform photoacoustic imaging at the maximum absorption wavelength of the dye before, during, and after the injection in the neck region. We can observe that the photoacoustic signal of IR783 halts in less than 2 minutes, while ICG clearance was only slightly faster than that of ICG loaded on the solid lipid nanoparticles. But blood elimination of gold nanorods was much slower than the other contrast agent. We then move the transducer in the liver region and we look at the liver uptake. So we observed that higher 783 showed a very low photoacoustic signal at all the time points observed, while ICG showed a maximum signal at 20 minutes, which remains stable up to 1 hour. ICG loaded in solid lipid nanoparticles showed a slightly later uptake compared to ICG. So we have a maximum signal of ICG solid lipid nanoparticle at 40 minutes after the injection, while gold nanorods showed the maximum signal after 4 hours, which remains stable up to 24 hours. This data suggests that IR783 is not a good photoacoustic contrast agent. This is probably due to its high fluorescence quantum yield after plasma protein binding. We then perform optical imaging experiment to validate the results obtained with photoacoustic imaging. So we perform optical imaging after 2 hours and 24 hours post-injection. We can observe that the signal of ICG and ICG loaded on the solid lipid nanoparticles is high at 2 hours after the injection, but while at 24 hours the optical signal decreased by 10 times. IR783 showed a strong signal after 2 hours and 24 hours post-injection. For gold nanorods, the signal was weaker than the other dyes, so it's very low compared to the other dyes. We also, after this preliminary experiment in vivo healthy mice, we perform tumor uptake experiment in tumor-bearing mice. So the tumor uptake was validated in mouse model of subcutaneous glioblastoma U87MG. So we inject ICG-RGD, ICG solid lipid nanoparticle, gold nanorod, and the quencher molecule in U87MG tumor-bearing mice, and we look at the tumor uptake. So these are the results regarding the gold nanorods. We observed that a weak signal is detected at was observed after 15 minutes post-injection, but no photoacoustic signal was detected in U87MG tumors at later time points. These are the results concerning the quenchers, so the asymmetric cyanide. That showed a low but stable uptake in U87MG tumor. This data suggested the further investigation on this class of PRET quencher for photoacoustic imaging, maybe for some targeting, because this is an untargeted molecule, but we can target it with some vector and perform molecular imaging, photoacoustic imaging. Concerning the ICG-based photoacoustic contrast agent, from the in vitro evaluation in agar phantom, we observed that the signal of ICG Selen was double compared to the signal of ICG or ICG-RGD. For this reason, we injected half dose of ICG loaded in the nanoparticles compared to the other two dyes, in order to have a similar signal for all the three contrast agents. So these are the results of ICG, ICG solid lipid nanoparticle, and ICG-RGD. They showed high photoacoustic signal in tumors already after 15 minutes post-injection, suggesting that in this early vascular phase, all the contrast agents diffuse similarly. But 1 hour after the injection, ICG showed a 70% signal reduction compared to the signal after 15 minutes, while the signal of ICG Selen was high up to 1 hour. And this is, so we successfully improved the biodistribution and blood clearance of ICG, loading ICG in the solid lipid nanoparticles. The signal of ICG-RGD was elevated up to 24 hours after the injection. Up to 24 hours after the injection. This is likely due to an integrin-mediated accumulation of the probe. So in order to validate the results, these results, we inject ICG-RGD also in A431 tumor-bearing mice. That is a tumor model characterized by a low alpha-V beta-3 expression. So ICG-RGD showed a different behavior after injection in A431 and in U87MG tumor-bearing mice. The photoacoustic signal of ICG-RGD in A431 tumors rapidly decreased, while the signal of ICG-RGD injected in U87MG remained stable up to 24 hours. In order to confirm these results, we perform optical imaging experiment after 2 hours and 24 hours after the injection. We can observe from these results that there is a nice signal for all the injected contrast agent after 2 hours post the injection, but after 24 hours post the injection, the signal of ICG and ICG solid lipid nanoparticle injected mice was low. But we can observe that there is a nice signal both in A431 and U87MG bearing mice, and this is due to a slower blood clearance of ICG-RGD compared to the free ICG. But we can also observe that the signal derived from U87MG is greater than the signal derived from A431. This confirms the results obtained with photoacoustic imaging. To have more information about probe biodistribution in the tumor, cross-section were obtained from U87MG tumors injected with ICG and ICG-RGD, and we can observe that a weak signal was derived from the tumor injected with ICG, while a strong signal derived from the tumor injected with the ICG-RGD, confirming imaging findings. In this magnified image, we can also observe that the signal derived from ICG-RGD seems to be from the parenchyma of the tumor and not only from the vascular phase. So, in conclusion, we evaluate different photoacoustic contrast agents, and regarding small molecule near dyes, we evaluate the asymmetric higher dye quencher that showed a low but stable uptake in U87MG, and so we can, in the future, we can perform further investigation of the class on this class of contrast agent for photoacoustic imaging application. The gold nanorods show a strong liver signal, but the signal in the tumor was very low. Regarding the ICG-based contrast agent, we observed that the ICG solid lipid nanoparticles were optimized for photoacoustic application, increasing the amount of ICG loaded in the nanoparticles, and they showed a good and a greater photoacoustic tumor enhancement compared to the free dye, and also a lower blood clearance. The novel targeted agent ICG-RGD reported non-invasive on-tumor heterogeneity and showed a strong signal up to 24 hours post-injection. So these appear to be a valid starting point for targeting strategies for molecular photoacoustic imaging. I would like to thank all of you for your attention and VisualSonics for the opportunity to share my results.
Great. Thanks so much, Dr. Capozza. That was a great presentation. I'd just like to remind everybody that we will be taking questions now. So if you've got any questions, please just type them into the questions box, and we'll try and get through some of those questions. I'm going to start by asking a couple here. First, just generally, why use multimodal agents in this case? Why is this important for cancer imaging?
Yeah, we can say that you mean the cell-deep nanoparticle here, or not?
Yeah, I guess just multimodal agents, so being able to see them with more than just photoacoustics alone. So photoacoustics, yeah.
Yeah, in this case, I think that the optical imaging gave us the opportunity to have information about the whole body mice, so also the biodistribution, so we can study the biodistribution of the our imaging probes in order to have information about the target organs and in order to validate the results obtained with photoacoustic imaging. So in this preclinical approach, I think that having a probe that can give you signal in both in optical imaging and photoacoustic imaging is a great opportunity. So, for example, for the gold nanorods, we don't have any signal in tumor via photoacoustic imaging, but they don't give us any signal in optical imaging. So we can say if this gold nanorods goes in the tumor or not. That's I think that this is an important case, and it's important to have information from optical imaging also.
Okay. Yeah, great. So a question that's come in that's related to that is, and you mentioned, using another modality to validate some of these results. A question is coming in saying, in the U87 model, you're talking about uptake in the tumor, but they're saying it seems that the ICG RGD is localized around the tumor in the rim, around the rim of the tumor. Do you want to just give us some explanation of that, if possible?
Yeah, of course, we select two tumor models that have different alpha V beta 3 expression, but we also know that the alpha V beta 3 is also expressed by the neoangiogenic vessels, and in these xenograft models, I think that an important component of alpha V beta 3 expression is also from the vessels of the neoangiogenic vessels. So we can observe a difference in U87MG and A431 because they express different, they have different expression of alpha V beta 3, but we also have to take in consideration that we have also other vessels from the mouse.
Okay, great. Yeah, and just in my experience as well, I've seen there is this rim effect with these tumors where there may be neoangiogenesis happening around the rim of the tumor, and therefore, as you mentioned, you get this distribution. But I think also because that's sort of where these larger feeder vessels are, you tend to get more distribution around that rim as well. There's another question here. Let's see. Do you have an explanation for why untargeted QC1 went into the tumor when the structurally similar dyes, like ICG, did not?
It's probably due to the albumin binding. So if with what we think that happened is that when injected to puncture, it probably binds to albumin, and so via EPR effect we can have an untargeted accumulation in the tumor, so passive targeting in the tumor. ICG, of course, binds to albumin, but is characterized by very fast blood clearance, so it's difficult to see ICG after, for example, 1 hour in the tumor.
Okay, so you're saying that the clearance of ICG is likely just responsible. It's cleared so much faster that it just doesn't have the chance to have this exactly. Non-specific accumulation versus QC1.
Yeah, of course.
Okay. Is there any reason why you see such a long circulation time of the gold nanorods versus, you know, these other dyes and things?
I think that it should be due to their dimension, because the gold nanorods are metallic nanoparticles, so they and they are pegylated, so they have a very slow blood clearance, and they accumulate in the liver. So it's all these features are typical of nanoparticles, and in this case, metallic nanoparticles. Yeah. While other molecules like ICG have a faster blood clearance, as we already thought. Moreover, when we inject ICG in the mouse, they bounce to albumin, and this increase their fluorescence quantum yield, and we expect that this decrease the photoacoustic signal. So all these factors affect the blood clearance that we measure via photoacoustic imaging.
Okay, but I mean, your solid lipid nanoparticles were also pegylated, right? Why do you think the blood clearance of those was faster than the gold nanorods?
Yeah, of course, this is a question that we already asked. They are totally different nanoparticles. So the gold nanorods are metallic nanoparticles. The ICG are made of phospholipids. Moreover, so I think that this can affect the blood clearance. But moreover, the loading of ICG is not could be not as stable as the gold nanorods, and this can affect, of course, the blood clearance.
Okay, I see what you're saying. So related to that, are your solid lipid nanoparticles, are those biodegradable? So for example, if they, you know, get into the tumor in a non-specific way, will they biodegrade and sort of release the free dye at that point?
We didn't study it. We didn't make any experiments about this. So I really can't answer to this question. But what I think that they are biodegradable, of course. Unfortunately, we don't have stability in vivo of these nanoparticles, of course. Okay,
yeah, understood. It's a complex question. Yeah. It's a difficult question to answer. I understand, but okay. Another question. There's somebody asking a question about the recommendation for the best agents for visualizing metastatic tumors. So, in your experience, you know, would your particles that you've created here be good for trying to image metastasis?
No, actually, we don't. Of course. No, actually, we don't. We just try these nanoparticles in xenograft. That is, of course, more superficial than I think metastatic. So no, I don't know if we can do it. Actually.
Okay, but I mean the reason for developing these kinds of agents is one of the applications to be able to, you know, image sites of metastases. So if you have a targeted particle, you know, if you know where the tumor is, you can see the distribution of the molecule that you're interested in. But is one of the applications as well to be able to develop these types of particles so you can just inject them systemically and be able to detect metastasis?
Of course, we observed that this is more the aim of ICG-HDD, because we observed that the melanoma overexpressed the alpha V beta 3, so we can do something about, we can increase the specificity of diagnosis of melanoma via using ICG-HDD compared to the ICG without any target.
Okay. Okay, great. Thanks. That's actually one more question about the optical imaging. I noticed, and I may need you to go back to that slide because I can't exactly remember, but it seemed there was actually a lot of signal in the kidneys. and usually ICG is cleared through the liver and then through the gut. So oh, here we go. Yeah, thanks.
Yeah. Okay.
So is there a reason that the targeted ICG seems to be really highly sort of taken up in the kidneys? Do you have an explanation for that?
Yeah, we think that, of course, the ICG and the ICG-RGD, we expected a totally different biodistribution because at the end they are different molecules. Of course, the dye part is the same, but the RGD binding could affect the clearance of the molecule, of course. And yeah, we was surprised by this kidney clearance, but yeah, we think that maybe because it's a small molecule, we can have some clearance from kidney instead of liver.
Okay, interesting. So this is just a difference in biochemistry and the resulting sort of pharmacokinetics and biodistribution of the.
Yeah, also because, for example, after 24 hours, the signal from the whole mouse derived from ICG is lower compared to the signal derived from ICG-FGD. So it's in this case we have different biodistribution, different blood clearance also, because you can see that in the case of ICG-FGD injected mice, the whole mouse gives signal compared to a greater signal compared to the ICG.
Right. Did you take a look with photoacoustic imaging at any biodistribution? So did you take a look at the kidneys of these animals?
No, we don't. No, we don't. In vitro, you mean, or ex vivo?
In vivo, because I guess at the different time points you could also, you have the organs excised here obviously, but at the different time points you could look in vivo and just see differences in biodistribution potentially.
No, we don't. Actually, we don't. No.
Okay. All right. Well, thanks so much, Dr. Capozza. Thanks everybody for. Thanks everyone for attending. If if you do have more questions, please send us an email. You can go to our website to find our contact information. Is www.visualsonics.com, and just go to the contact page and send us your questions. I've got a couple short slides to show here. We've got a couple of promotions that we're working on right now. One is the Vivo micromarker giveaway. So if you're interested in doing targeted molecular imaging, we've got ultrasound contrast agents. So these are phospholipid shell bubbles with a perfluorobutane core, gas core, and they're streptavidinated, so you can put biotinylated antibodies on them and do targeted imaging within the vasculature. And we're doing a giveaway of these bubbles right now. So if you have a targeted imaging study that you're interested in exploring, please submit via the website here. You can go to the link at the bottom of the page there. Submit your idea for how you would use the targeted bubbles for your study, and we're evaluating them here, and you could win a supply of bubbles. I just also want to point out some resources we have on our website. If you go to our Vivo blog, you can see how these bubbles can be used for molecular imaging. So again, these are FDA-approved bubbles that Bracco makes that were used in a study for detection of essentially liver cancer. So you can see how they're being used in the clinic and how you might be able to apply them to your preclinical models. I also want to point to our April 2016 webinar. So if you go under our resources page on our website under webinars, the data that Dr. Capozza was presenting here, this is sort of a workflow of how to use the Vivo laser system, the system that she was using to gather this data. This is how to use this. It's sort of like a tutorial of how going through a molecular imaging study for cancer research, looking at microdistribution and pharmacokinetics, very similar to the kinds of things that Dr. Capozza was doing, using different particles in this case. But this is just a really nice sort of walk through, stepping through each step of the study, from characterizing these agents in vitro to going in vivo, doing the multispectral imaging, doing the spectral and mixing quantification, and all of that. So if you want the really sort of in-depth details of how this study or these types of studies are conducted, you can check out that webinar. Again, it's under our resources page under the education webinars. That's an April 2016 webinar. Finally, although Dr. Capozza was using the Vivo laser, we do have a new system, which is the Vivo Laser X. This was launched back in February, and there's a few, and you'll notice right away it looks quite different than the Vivo laser system. It's really this multimodal system for doing co-registered multimodal imaging. You get anatomy, function, molecular information all in one platform. It's got a touch screen for really smooth workflow. It can be customized for a particular user or study. We have new laser technology for faster, more sensitive acquisition. We also have an expanded wavelength range. So in addition to the 680 to 970 nanometer range, we also have the 1200 to 2000 nanometer range. And we've customized the light delivery. So on the probes that Dr. Capozza was using, we integrate the light into the housing of the transducer and permanently. In this case, we're taking our transducers and we're able to snap a jacket over top and change the different fibers to get either a wide field of view or a narrower field of view with increased depth and sensitivity. So you can use the illumination that suits the application. We've talked a lot here about molecular imaging, but there's also applications for cardiovascular applications, looking at things like ischemia, oxygen saturation, that kind of thing. And finally, you know, again, Dr. Capozza is talking pretty much about just the photoacoustic imaging aspects, and she's of course showing these images co-registered with high resolution anatomical detail with the ultrasound. But it should also be noted that, you know, there are many other tools on the system as well. So going from, you know, this in-depth, you know, multispectral imaging for doing the molecular imaging, right down to just doing plain, you know, tumor sizing, seeing the anatomy, also doing hypoxia and ischemia measurements. There's blood flow and perfusion measurements using the microbubbles as well, and then targeted imaging using the microbubbles. So there's lots of features and ways of quantifying your data using the system. Again, please reach out to us if you have any questions about the technology. Thank you again, Dr. Capozza. Thank you everyone who's attending. And again, we've recorded this webinar and we'll be distributing it within the next week or so. Thanks very much, everyone. Have a great day.